Hello and welcome to the Internal Medicine Grand Rounds. Please take a moment and sign into today's event using the QR code and activity code on the screen. The information will also be entered into the chat. There will also, in order to receive credit through our CME system EADS, you will need to log into your learner profile, complete the evaluation for this activity, and any other post-tests or surveys if available for additional credit. Lastly, at the end of the presentation, you will have an opportunity to ask questions. And today we have Dr. Lake, who will be introducing our presenter, Dr. Barr.
Great, thank you. So welcome to Grand Rounds, and I'm very excited today to be able to introduce Dr. Katie Barr as an outside guest speaker. Dr. Barr is Assistant Professor of Medicine at the University of Pennsylvania School of Medicine and Director of the Viral and Molecular Core for the Center for AIDS Research at UPenn. And she's a clinician researcher whose lab studies novel approaches to HIV prevention and cure, including monoclonal antibody development. And monoclonal antibodies are so relevant currently to everyone's practice inside infectious diseases and outside that I think we'll hear a great talk. And with that, I'll turn it over to you, Dr. Barr.
Thank you so much. I really appreciate the opportunity to speak and the invitation. And I hope today, let's see if I can go back. I hope I can convince you that broadly neutralizing antibodies or monoclonal antibodies that have broad and potent activity against HIV are relevant for HIV prevention and treatment moving forward. So to start with, for those of you who don't spend all of your time thinking about HIV, it gets a little bit less attention given all the other various health concerns in the world. But it's important to remember that each year, about 1.5 million people are newly acquiring HIV. And this is a decrease from past. This is about 30% down from where we were about 10 years ago. But this is still a huge number of new infections each year. And this is more than the number of people that were able to start on antiretrovirals each year. So we are expanding the pandemic each year. In addition, we have almost 40 million individuals living with HIV. There are about 40 million people who have died from HIV, and each year the number of people who continue to live with HIV expands as well. And so clearly we need to identify new ways and novel approaches to prevent and to treat HIV. So the US government has an Ending the HIV Epidemic plan for the United States, which also applies to the world. And 2 of the 4 major tenets of this approach are to treat HIV quickly and effectively, and then to protect people at risk from HIV. And so my hypothesis here and the premise for this talk is that broadly neutralizing antibodies, or monoclonals against HIV, can contribute to novel approaches to both treat and prevent HIV. So let's talk for a minute about antibody responses in HIV, because when we think more broadly about infectious diseases, it's clear that antibodies are very important in clearance of infection, in prevention of infection, in correlates of vaccine protection, etc. However, with HIV, for various reasons, the antibody response is always too little, too late. And that's partly because the virus is very well protected from neutralization, and the virus attacks helper CD4 T cells that are important for developing a robust antibody response. But what we end up seeing is that our first antibodies against HIV develop early, but they tend to be cross-reactive antibodies that were originally arising against gut pathogens or gut microbiomes, and they don't do much to curb the HIV virus replication. The first neutralizing antibodies, which are strain-specific, meaning they tend to attack the actual virus establishing infection, but not necessarily viruses circulating broadly in the community or even viruses that will evolve later in that individual. So they're very strain specific. They can be potent, but they also are quite delayed, and they don't actually arise to have neutralization properties until somewhere in the 2 to 4 month range. Now, in a small subset of individuals, and this number varies depending on how you define antibody responses, but between 5 and 30% of people living with HIV actually go on to develop very high titer cross-reactive antibodies, or broad and potent neutralizing antibody responses. And these are the types of antibodies that we like to use in a monoclonal form for, as an intervention. But it's important to realize that the virus is always faster than the immune response in HIV. And so even in individuals who develop these antibodies, their own viruses have previously rapidly escaped from those antibodies. And so they're all resistant to whatever antibodies are present at that time. So the concept behind using bNAbs as interventions is to sort of change that time scale and take away the ability of the virus to rapidly escape to the response by using it before the virus can escape, and at very high quantities. So if we can take highly effective antibodies that potently neutralize and have effector capacity against the relevant viruses, and we give them at the right clinical juncture before the virus has time to evolve resistance, in theory, this could be very effective for preventing infection, for suppressing viremia, or for clearing infected cells. So that's the premise behind using bNAbs. So let's talk about a brief history of how bNAb discovery evolved. So it wasn't until decades into the pandemic that scientists actually recognized that humans had the capacity to even generate these broad neutralizing antibodies. And the first generation of these broad antibodies were identified in the 90s. They attacked multiple distinct epitopes on the HIV trimer, as shown here on the left, but they were of relative modest breadth and potency compared to what we use right now. But they were really serving as a proof of principle that the human immune system could generate broadly neutralizing antibodies. So if we fast forward about 15 years, lots of enabling technologies, including large patient panels, high throughput neutralization, B-cell cloning and probes, this allowed the field to identify multiple broad and potent monoclonals and gave us this possibility of using passive immunization, and that we could take these identified antibodies and use them as treatment or as prevention or as a curative strategy. And so today we currently have more than 7 distinct classes of antibody targets that can be used for bNAbs. And we have at least 4 distinct classes that are in advanced clinical development, where we've had clinical trials in humans for prevention or treatment. We also have a group of engineered monoclonals where these antibodies have been modified to have longer half-lives or better effector function, or to have multiple specificities within a single antibody. And together, this represents a real opportunity to use these antibodies for different approaches. So let's start with prevention. How can we use bNAbs for biomedical HIV prevention? So if we think of what we have available to us right now, we really have a wide, what we call prevention toolbox, incorporating many different strategies. We have antiretrovirals that we can use as pre-exposure prophylaxis. And certainly over the past 10 years, we've seen that oral PrEP, or Truvada, for example, can be highly effective when it is used. We also have long-acting injectables that were just recently shown to be superior to oral PrEP. And these long-acting injectables are now just very recently approved by the FDA. And we're starting to see how we can roll these out in larger communities. Obviously, a vaccine would be incredibly impactful, but that research is still ongoing. So how might bNAbs have a role in this context for HIV prevention? Well, I think one of the strongest rationales for using antibodies when we already have very effective small molecule based prevention is that this is a distinct mechanism. And therefore, if we have a failure on antibodies or breakthrough infection, we're unlikely to acquire resistance to the foundation of our standard of care treatments. So what I'm showing here on the left are study results comparing oral PrEP and long-acting injectable PrEP CAB is the long-acting injectable, and TDF-FTC is the oral prep. And what you can see, I hope, is that there were very few new infections on this injectable cabotegravir. And in fact, this was favored compared to the oral prep. But what we saw, even in this very effective clinical trial use of this, was that there were 12 incident and 4 baseline infections detected in this study. And many of these individuals acquired virus that was resistant to cabotegravir, which is sort of the backbone of our HIV treatments right now. And so the goal of using antibodies would be to avoid developing resistance to these really important drugs that are needed should an individual acquire HIV. So in general, the advantage or the rationale for using bNAbs are that they are long-acting, they are very non-toxic, and have a high therapeutic margin. We can give high doses without worrying about toxicities. They have reliable pharmacokinetics across populations, meaning in infants and in pregnant women or in pregnant individuals and in people with renal or liver dysfunction. We are able to monitor and use these safely. There's also the potential benefit that these antibodies can engage with the immune system of the person being prescribed, and that could be immunomodulatory. In HIV, we like to provide our clients with as many options as possible. So this provides another option for prevention. And then I think it's important to recognize that these antibodies may have specific advantages in key populations. For example, in pregnancy and in breastfeeding infants. This is a relatively short time frame, but 1 or 2 long-acting, broadly neutralizing antibody injections could be very effective in protecting during that specific time period. In adolescents, when drug resistance and adherence is a huge issue, these long-acting formulations could be beneficial. And certainly in individuals who have multi-drug resistant virus, this could be a new lease on effective therapy. So we've tested antibodies in the animal model, the non-human primate system, multiple times. And in general, it's been shown that if you give the right amount of antibody against a sensitive virus, these antibodies are extremely efficacious in preventing infection. So what I'm showing here is data from a meta-analysis of 18 different studies where they found that at high doses or at the right doses, these antibodies were highly effective. But the important thing that they recognized was that it actually takes fairly high levels of these antibodies. And so we need to get quite a bit of antibody into the right places in order for these to work, although they can be quite effective. So I just want to mention that the prevention in mother-to-child transmission has also been extensively studied in the non-human primate model. So on the left here, I'm showing a study of pre-exposure prophylaxis, whereby infant rhesus macaques were given doses of 2 antibodies, and then they were given multiple challenges of this virus orally to reproduce the concept of breastfeeding related transmission. And what they showed was these antibodies were extremely effective in preventing infection in this model. They've also tested post-exposure prophylaxis. So asking if we give the antibodies days after the exposure, can we also prevent infection? And again, they showed that at least at 30 hours, these antibodies were highly effective. And at 48 hours, there was some benefit as well. So we can fine tune the timing and the doses needed within this animal model. But as we all know, the models are not perfect replications of human data. And so I think it's important to emphasize how these trial data from non-human primates, or the best animal model possible, don't perfectly translate into clinical trials or real world use. So in our non-human primate studies, we use very specific virus challenges, and these viruses have been extensively characterized so that we know how infectious they are, we know what the sensitivity to the antibodies is. Whereas in a human situation, we don't really know what the virus challenge is going to be. We can estimate what kind of viruses are circulating in the community, but we don't actually know what's going to be the specific virus that any individual is going to be encountering. In addition, in the animal model, we have a defined number of challenges with very specific conditions. We're giving the dose IV or as a mucosal challenge. Whereas and we know that there aren't any concomitant conditions that could increase the risk of transmission. No sexually transmitted infections, no assault, no other medical conditions. And all of these things are real factors in the real world and clinical trial use of these antibodies in humans. In addition, these animals are pretty adherent to their study treatments, whereas we all know human behavior and real world conditions being what they are, nobody is able to be perfectly adherent to any sort of treatment or study protocol. And finally, I think this is another important concept. When we do an animal study, we know exactly when we're testing to see whether infection or whether breakthrough infection has happened. But in human trials, we really can't test people very regularly because we want them to be able to live their lives. And so we almost always have a delayed sampling post-acquisition, which is harder then to disentangle exactly what happened to lead to success or failure. So let's move on to the human trial of broadly neutralizing antibodies, the antibody-mediated prevention study. So these two studies actually are the only large-scale BNABs for prevention studies that have been conducted in humans. They were published just last year. And briefly, this is an enormous endeavor where through two studies in the Americas and in sub-Saharan Africa, looking at men who have sex with men and transgendered women in the Americas and cisgendered women in sub-Saharan Africa. Again, looking at over 4,500 participants, these individuals were given either a low dose or a high dose of VRC01, which is a moderately potent broadly neutralizing antibody, or placebo. They're given monthly doses over the course of 20 months and then followed each month to determine if they acquired HIV. All of these individuals were deemed at high risk, and so they were followed pretty closely. Unfortunately, what we saw in this study is that there was no overall significant protection. So in the treated arms, there was an average or an annual incidence of 2.35 infections per year, whereas in the placebo arm, 2.98. So there was a trend, particularly in the highest antibody level receiving group, but it was not statistically significant. But if they had pre-specified a secondary analysis to ask about viruses that were inferred to be highly sensitive to this antibody. And what they showed in this analysis, and here we're looking at the top graph, that lightest gray line, which shows IC80 less than 1 microgram per mil. And what they showed here is that, in fact, if they looked at viruses that were sensitive to this antibody, there was actually a striking protective efficacy. So 75% estimated prevention efficacy against sensitive viruses. And so this is obviously not the home run we were hoping for, but it does provide optimism that a more broad or potent BNAB could provide strong protection because we'd increase the number of those viruses that were sensitive and therefore would be protected. Since this trial, there's been a ton of analysis of the data, and one of the lines of analysis is to estimate exactly how much antibody do we need for protection. And so a group of statisticians led by Peter Gilbert came up with this term called PT80, or the predicted serum ID80 titer. And this involves both the amount of antibody in the blood and the relative sensitivity of the viruses that an individual is exposed to. And if you have those two pieces of information, they have pretty good modeling data suggesting what relationship you need to prevent infection at a very high level. And so these information provide us a goalpost for what we need for when we ultimately generate a vaccine or when we use BNABs as passive immunization. So as of right now, the field is continuing to study prevention. There are many new antibodies that have greater potency than VRC01 that are available. And there are many studies in distinct groups, including at-risk infants and children living with HIV. And so I think there's going to be a lot of study data coming out, in particular just in the next couple months, as CROI rolls around in children, but in other study groups also. In addition, there's work to try to make these antibodies higher concentrations and longer lived through vector delivery, etc. And I think all of those things are going to translate to a better opportunity for prevention. But one question that I think is a big deal in the prevention community's mind is, how do we do the next clinical trial? because as I hope I made clear, those AMP studies were enormous expenditures in time and energy and dedication from the participants. And so the question is, do we just move forward with another large prevention trial? Or is there some sort of smaller trial that we can do as a surrogate for preventive efficacy? And so right now there's at least one attempt to try to do a surrogate study, sort of an analytical treatment interruption for prevention, where we actually test combination of antibodies in people living with HIV who interrupt their therapy and switch to these antibodies and ask whether that antibody can suppress virus. And if it can suppress virus, we hope that is a surrogate for the ability for those antibodies to also protect. So right now there's a trial in development called ATI for Prevention. It's aimed to occur in several countries in Southern Africa. It will involve 60 people living with HIV who are currently suppressed on antiretrovirals. And these individuals will either get a combination will either get a combination of antibodies or placebo and then interrupt therapy. And hopefully this data will allow us to know whether those antibodies would be good to support in a larger prevention study. Okay, let's move to treatment. So HIV treatment has obviously come an extremely long way in the past 30 years, and we now have very effective and well-tolerated daily oral antiretrovirals that allow individuals with HIV to have very healthy, long-lived lives. We also have long-acting antiretrovirals, multiple formulations now that are available, and this is another opportunity. So our small molecule antiretrovirals are quite effective. So why do we think that antibodies could potentially have any role in this context? For me, I think it's important to think about what do we mean by HIV treatment. And I think in my mind, it's a continuum. So on one end, we want to suppress virus. We want to prevent the immunopathogenesis of this virus and allow people to live really healthy, functional lives and not be at risk for transmitting the virus to anyone else. But we also want to think about other things that we can do with treatment. Can we engage the immune system to better suppress virus on its own? Can we clear virus-infected cells that constitute the reservoir, which is the barrier to HIV cure? Small molecule antiretrovirals are unable to do anything except prevent the next round of infection. But antibodies, because they have these other properties, have the ability to both treat and to work towards these combination cure strategies. And so I think for me, that's the major benefit that antibodies have. So they clearly offer long activity and low toxicity, high therapeutic margin, and lovely PK, but these theoretical benefits of clearing reservoir cells and inducing an immune response via the vaccinal effect are what I think is the most exciting part of antibodies for treatment. So let's just dig into that for a second. So on the left, we're showing a cartoon of reservoir reduction via effector function. And so as we all know, antibodies, by identifying virus-infected cells, can help the immune system clear this reservoir. And in theory, this could be an important part of antibody-dependent cellular cytotoxicity, phagocytosis, or other means of clearing these reservoir cells. In addition, a highly effective antibody can be an important immunomodulatory agent. And so there's this concept of the vaccinal effect, which is well documented in cancer, whereby if you have large quantities of virus and antibody binding together to form immune complexes, those immune complexes engage with antigen-presenting cells, and they lead to better responses against that antigen. And so we could see improved CD8 T cell and improved B cell responses against the virus with an effective antibody. So these are lovely, have been described in other contexts, but are not that well delineated in HIV. And therefore, it's a real priority for the cure field to understand if and when these properties can happen with antibodies. So again, the roles for these antibodies are both virus suppression and these other effects. But let's talk for a minute about what have we shown about durable virus suppression. So beginning in about 2010, these antibodies were developed to the point where we could start to test them in animal models. And so going back to our non-human primate data, we showed in viremic animals about 10 years ago that a single dose of antibodies could reduce viremia transiently, and this looks very similar to what we see with a single antiretroviral agent. So we saw good antiviral effect. Shortly thereafter, animals were tested with 2 antibodies, and the combination of antibodies targeting complementary epitopes was able to provide even more durable suppression. So we see longer periods of time without the virus coming back, and essentially waiting until the antibodies wane from the system before virus rebound. So again, this is good proof of concept that these can be used for humans. So first published in 2014 and 2015, we see that these antibodies can show similar effects in people living with HIV. So this is a trial in which viremic individuals were given a single dose of a CD4 binding site targeting antibody, 3BNC117. And what you can see from these blue lines and the median red line is that virus dropped more than a log. It stayed that way for a few weeks until the antibodies waned, and then virus came back. So again, we see this transient effect in viremia from a single antibody. So next, the field moved on to try what we call treatment interruption, ATI studies, or switch studies, where basically people living with HIV suppressed on antiretrovirals are given an antibody and have their antiretrovirals stopped. And we ask if and how long that antibody can suppress viremia. So this is a study that I was involved with testing VRC01, that same modestly potent antibody that was used in the AMP studies, but this was used in individuals stopping their therapy. And there were 2 trials, one by the ACTG and one by the NIH here, shown in green and yellow. And you can see in this Kaplan-Meier curve, the time that virus was suppressed was slightly greater than that blue line, which is historical controls. Now, a more potent version of a CD4 binding site antibody, 3BNC117, had a similar approach, and what it showed was even longer periods of time of maintaining virus suppression at a treatment interruption. And so these, again, showed that in this switch study context, we can suppress viremia against some viruses. However, we know that people who had resistance to these viruses in their virus populations had very early rebound and did not seem to be successfully treated. So this is just a brief introduction into how we did those analyses. This is the time after treatment interruption, and then the viral loads as they came back, as the antibodies waned, or as the virus broke through high concentrations of antibodies. And what we can see in some of these individuals who rebounded quite early is that if we characterize the viruses that were pre-existent, and here on the right, I'm showing a phylogenetic tree of pre-existent viruses in black, and then rebound viruses in red and orange, with the inhibitory concentrations, IC50s, on the right. And although this may be a bit obtuse, my point here is that what we showed is that in people with very early rebound, they in fact had viruses that were already resistant to this antibody, and that was the correlate of of early rebound or no effective response. So we know that baseline resistance is extremely important in understanding whether these antibodies work or not. Okay, so the field has moved on, thankfully, and we've tried combinations of more potent antibodies. So this is a study conducted by the Nussensweig group out of the Rockefeller, and what they showed on the right here is that on a total of 11 participants, this blue line is the Kaplan-Meier curve for how long they maintained virus suppression. And if you compare that to the red line of a single antibody, or the black line of historic controls of people who had no intervention, you can see many weeks of durable virus suppression with this combination BNAb approach. And so post hoc, the group went back and did extensive analyses of pre-existing viruses, and they in fact were able to determine that if people's virus populations were sensitive to these antibodies, they in fact had many, many weeks of virus suppression, a median of 23 weeks, almost 6 months. Whereas if people had highly resistant variants, they had very short periods of time of virus suppression. So this resistance to these antibodies is clearly essential to understanding if and in whom they're going to work. So screening for BNAb resistance logically is a very high goal for using these. We know that with any individual, there's a diverse population of viruses. The laboratory methods required to identify all of these viruses and test them are extensive and expensive and time consuming. And so the goal was to see, could we develop a CLIA certified assay that is able to be applied to available specimens, plasma, cells, and that is feasible and predictive of bNAb efficacy. So this is a recent project that we engaged in where we tested the one CLIA certified assay. This is an assay called the PhenoSense bNAb assay. It's based on a validated assay that's used for phenotypic testing for drug resistance mutations. It can take virus from plasma or from PBMCs or cells collected in people who are suppressed on antiretrovirals. It clones out the virus envelopes, generates a swarm, and then you can test them in this high throughput cell-based luciferase assay to see what concentration of antibody prevents further infection. And so this is the one assay available to us. And so through the ACTG, we decided to do an assessment of whether this assay is reproducible, whether it's actually predictive of clinical outcomes, and whether it's feasible in the context of a clinical trial. So in this study, we looked at an antiretroviral study, A5257, and 65 participants who had samples from the plasma before they started antiretrovirals, and then PBMCs or cells after 1 year of virus suppression and 3 years of ART-based virus suppression. And then we tested these against 7 antibodies in clinical development. So what we showed to begin with is that the assay was pretty successful. We were able to generate and do the assay in 92% of the samples. And then you can see here, this is the range of sensitivities across each of these 7 antibodies for the 65 participants for each antibody. And what I can tell you is there's a substantial range, multiple 3 to 4 logs for each of these antibodies. The median and the specific sensitivities varies from person to person clearly, but the general distribution of breadth and potency really agrees with what had previously been published using large panels of virus isolates in the clinical development of these antibodies. So that was reassuring. So then we went on to test the specifics of the assay. And so we compared the values generated from the different samples, individuals' plasma before they started antiretrovirals, and then cells at 1 and 3 years of ART suppression. And I hope you can see that these distributions look very similar. And in fact, when we compare the values between samples, we can see they're highly correlated. And it really shows that from sample to sample, we can get the same results. Similarly, if we compare the measures of this assay and we look at the lab values that were generated in vitro for various studies, we see there's a strong correlation between sort of comparing apples to apples and like comparisons in these in vitro assays that take a little bit more time and energy versus this screening assay through Monogram. And so this is one study where we had just 5 samples and there's a very nice correlation, but this same correlation can be seen across multiple studies that we looked at. However, when we applied these baseline assessment retrospectively to 3 different studies of bNAbs at treatment interruption, unfortunately, there was not good prediction based on the continuous variable of the resistance of whether the antibody would be able to suppress that person's viremia for a period of time. And so unfortunately, the IC50 or the IC80 that was generated did not tell us how long or how effective that antibody would be in suppressing viremia. It suggests that even though this test is reproducible, it reproduces some lab values. First of all, we may not be sampling relevant viruses. We know the reservoir is huge and we're just getting 1 sample. And secondarily, it may be that the clinical efficacy of these antibodies is based on more than just the sensitivity. And so this may be one important component, but maybe not fully explain what's going on. Secondarily, this assay is fairly logistically challenging in that when we looked at the real world experience from 3 recent studies, the assay success rate was lower. We're not sure exactly why. And the median processing time was between sort of 1 and 2 months. And if you're trying to enroll a study that's already challenging, that can be a real impediment to study feasibility. So I think these are important characteristics to consider, as we definitely want a screening assay, but this may not be the best approach as it stands. All right, so we're back to suppressing antibodies being able to suppress viremia in people who are living with HIV on ART. And so again, with combination antibodies, we've had multiple studies recently showing they do a pretty good job. But we have not seen huge changes in the reservoir or in the immune responses of these individuals when antibodies are given at the time of ART suppression. And so one of the questions that arose recently is, can we give the antibodies at a different time point that may be more effective? And so one recent strategy is to use these antibodies at the time of ART initiation. So as an individual is viremic, there's lots of virus circulating, and they're just starting antiretrovirals, can we also give them antibodies? And that allows two things. One, it allows changing of the reservoir, and two, it allows immune complexes. So what I mean about the reservoir is that recent work has shown that the reservoir, or the group of HIV infected cells that persists through antiretroviral therapy and is the barrier to cure, this reservoir is largely formed at the time of ART initiation. So this was shown very nicely in a study by the Caprisa cohort, where they followed 9 women who were diagnosed an acute infection, followed through viremia over many, many weeks to years, and then characterized as they started antiretrovirals and were suppressed for years. And then they had their viruses from viremia, from their years of viremia, compared to reservoir viruses characterized years later. And what this study found was overwhelmingly these reservoir viruses most resembled the viruses circulating at the time of ART initiation. And to make a long story short, this is because the antiretrovirals stabilize the immune system, and they allow these infected cells to do what they're supposed to do and become long-lived memory CD4 T cells. And so because of that, the majority of the stable reservoir is actually founded by viruses circulating at that time point. The second important thing about ART initiation is that there's a lot of virus present just before you start antiretrovirals, and that hangs around for a little bit of time if we add antibodies to the mix. And so that enables these immune complexes, antigen in the form of virus and antibodies, to come together, engage with the host immune system, and maybe be immunomodulatory. So this concept of using antiretrovirals at treatment interruption has been tested in the non-human primate model, and what it showed is that giving antibodies at early infection at the time of ART initiation led to good immune responses, and then when antiretrovirals were interrupted later, as seen in these purple lines, the virus did not come back in the way it did when animals were just treated with antiretrovirals. And so this suggested that antibodies and antiretrovirals at that early time point could improve immune responses and potentially lead to durable virus control. So there was a study published just this past year that tested this exact concept in a human clinical trial. So this is the eClear study, and in my opinion, this is one of the most exciting things in the HIV cure world for quite a while. Maybe that's a low bar, but it's still pretty exciting from my perspective. So the eClear study was led by Ole Sjogard, who's an investigator out of Denmark, and it was largely conducted in Europe. Keep in mind, this was done in the middle of COVID, so there are some things that they weren't able to do that were part of their initial plans. But what they did was they had 4 arms in which individuals were diagnosed and then started on antiretrovirals. Some of these folks were early in infection, some were later, but at the time of ART initiation, they were either given just antiretrovirals, or they were given an antibody, 3BNC117, which is this more potent CD4 binding site antibody, or they were given a latency reversal agent, so something that's meant to make the virus come out of latency and show itself to the immune system, or they were given both the antibody and the latency reversal agent. And the hypothesis was that if we give this antibody at the time of ART initiation, we may be able to chip away at the reservoir better, and we may be able to boost host immunity for better virus control. So what they showed, first of all, is that when they looked at plasma viremia over time, people who got antibodies had a faster decrease in the virus in their plasma, and they had a faster decrease in the number of virus-infected cells. And in particular, they started to differentiate with people who, using that PhenoSense assay, had sensitive baseline virus, versus resistant baseline virus. And so in the individuals who had antiretrovirals antiretrovirals and were sensitive, they saw marked decreases in the number of infected cells over time. Then they looked at the CD8 T cell responses that were specifically targeting HIV. And what they showed was a greater number and a greater potency or greater functionality of these cells. So here they're comparing in orange our antiretrovirals alone to sort of darker blue on the left here, all of the antibody receiving individuals. And here in the more aqua color, antibody receiving individuals who were also sensitive to the antibody. And if we look specifically at those individuals, we see substantial increases in the number of HIV-specific or GAG-positive CD8 T cells. In addition, when they looked at the functionality, so in this case, they're looking at interferon gamma production, in the people who got antibodies and were sensitive, we saw marked improvement in the functionality or the cytokine production of their CD8 T cells. So finally, they were able to conduct a treatment interruption, an optional treatment interruption. And again, this was COVID, and so they made this part of the treatment of the study optional, and only individuals who wanted to engage in this could, and given various COVID restrictions, many people opted not to. But when they compared the individuals who received 3BNC 117 and were sensitive to the virus, what they showed is, again, this is a treatment interruption situation. So on the x-axis, we're talking about time after stopping their antiretrovirals. And again, this is years after the initial treatment. And then we're looking at the virus that comes back. And in green, or this yellowish color, these are the folks who were resistant to the antibody. And in the open bluish teal squares, you can see the folks who were sensitive to the antibody, in fact, had quite a change in their virus rebound, and their virus tended to be lower and looked like their host immune responses were better controlling it. And given the criteria they had for restarting antiretrovirals, many of these individuals, 4 of the 5, were able to stay off antiretrovirals because their virus was still quite low. And so this is a small trial, and this treatment interruption in particular is just a small cohort, but I think that these antibody at ART initiation study provides cautious optimism for multiple new trials testing broadly neutralizing antibodies and other immune interventions at this time point of ART and initiation, when the reservoir is vulnerable and when immune complexes are able to be formed to engage with the host immune system. Okay, so the final point I want to make is that as we endeavor to develop broadly neutralizing antibodies, the concept of equity is really important. And we need to think about that in each stage of our development. So as we are developing these antibodies, are we making sure that these antibodies have activity against viruses circulating in the whole world, and not just in the West, where we have predominantly clade B virus, but in Africa, where clade C and A and D are important, in Asia, when different types of A and AE viruses are important. So we need to make sure we have antibodies targeting the range of viruses. As we engage with our commercial drug system, where many of these antibodies have now been picked up as potential products, are we allowing the science to continue as these antibodies are now subject to patent law, etc.? So I think it's really important we let the science continue. In terms of clinical testing, are we performing clinical trials in the most important populations, those who need the intervention the most, or those who could benefit the most? And that includes the highest prevalence regions. Clearly, areas of sub-Saharan Africa are the most important for these prevention and treatment studies. Also thinking about important populations, women who are traditionally excluded from a lot of our clinical trials, pregnant and breastfeeding individuals, infants at risk and adolescents who oftentimes are way down the priority list for when things get tested in terms of interventional trials. And finally, as we develop these products, how can we make sure they're accessible to the whole world? We need to make sure these antibodies are cost effective and accessible. So this involves the obvious issue of manufacturing and licensing these antibodies, but also can we use innovative delivery methods or longer acting agents to get more bang for our buck and make this a truly cost effective and reasonable strategy when and if it rises to the point of meriting such consideration. Okay, so in summary, I'll thank you very much for your time, and I hope that I've been able to explain some of the important issues about broadly neutralizing antibodies for HIV prevention and treatment. And in my mind, the recent successes that we have include the fact that we now have combination broadly neutralizing antibodies that demonstrate efficacious virus suppression of sensitive viruses. And we can see this in many contexts. But I think the second part of this is we are developing new antibodies with modifications and better delivery methods to make sure that the range of sensitive viruses or individuals who could benefit from the antibodies available expand. I think some challenges clearly remain. First of all is how do we translate the suppression data to prevention strategies? It's not direct, and I think it's a major priority, but we need to be thoughtful about how we can move the prevention field forward, using the successes of virus suppression. The issue of resistance to these antibodies, both baseline and emergent on therapy, is really important. We need screening assays and development in that regard. We need better antibodies, and we need better combinations of antibodies to sort of mitigate that resistance. I think we need to speed up our manufacture and testing of these approaches because these things take a very long time, and we have, I think, a limited amount of time to do this work to really be as impactful as we can. But I am optimistic about this field because, in particular, I think the immunomodulation and the virus reservoir reduction seen with the eClear study and other similar studies that are going to come out very soon suggests that we may be able to harness the immune system in a way to improve virus suppression outside of what we can do just with antiretrovirals. And I think the next few months and years will be really exciting with multiple ongoing studies presenting their results and hopefully informing understanding of what is truly possible. Okay, thank you very much. And I'd be happy to take questions if there's time.
At this time, if you have a question, you could either partner with one of our chief residents at the LBJ Annex or the Memorial Hermann conference room. You could also unmute yourself, or you can place a question in the chat.
Katie, this is Roberto. Thank you for this wonderful talk, as always. I have a quick question. Something important that you're mentioning there is the manufacture of these VNAPs and trying to deploy it, particularly in the developing world. And what is your, input from that point of view about, making this available to a large population?
Yeah, Roberto, great question, and nice to hear from you. Always lovely to hear your voice. So right now, manufacturing is a clear bottleneck. We have a few groups who manufacture their antibodies. Most of these are Western, either United States or European groups, or international groups that manufacture antibodies they're developing, or companies that have purchased the right to these antibodies and are manufacturing them as future products. So that has led to challenges even to do the relatively small number of clinical trials that are being conducted right now. So when and if we get to the point that we have an antibody that we feel confident can do what we want it to do, and we need to make more, there's going to need to be an effort to really ramp that up. There are strategies that are being discussed, things like the Serum Institute of India and other organizations that are highly adept at making complicated medications, vaccines, antibodies in a generic format around the world. These sort of groups will be necessary to engage. But I think the very sticky business of dealing with patents and dealing with scientific ownership and the whole sort of monetary side of things is going to be challenging, and it's going to take a lot of advocacy from, I think, HIV scientists and advocates, as well as other groups, to make sure that we get these antibodies made in a cost-effective manner so that we can use them where they're really needed.
And I have another question regarding, you mentioned very well the need for this combination, and many of the studies have been done, to my knowledge, up 2 or 3 VNAbs. And even with 3 VNAbs, we have been seeing a rebound or after a concentration of the VNAbs, of sometimes rebounding with the resistant virus. What will be the next approach? Do you think that we will need 4, 5? What will be the combination, or developing a different type of VNAbs that the same VNAb will have more than one target?
Yeah, great question. Yes, so I think we made a huge jump when we went from sort of modestly potent single antibodies to 2 very potent antibodies in combination. And I don't know that we've seen a huge leap when we add that 3rd agent yet. And I think that's both to do with the specific antibodies that we're using. We've really not gotten to the point where we have a group of 3 antibodies in which, for example, the average clade B virus is sensitive to all 3 of them. And usually what we're doing is hedging our bets, thinking that 2 of the 3 will be potent. But I think with the continued development of antibodies and the continued understanding of which antibodies we partner with each other, we'll be able to get to the point where going from 2 potent antibodies to 3 potent antibodies will make a difference. Right now, we're kind of going from 2 potent antibodies to 3 antibodies, of which 2 are potent. So it's not a huge increase. There are some sort of engineered antibodies. For example, the ECD4-IG, which is a engineered antibody that has incredible breadth and potency. And when we get those sorts of antibodies, those in combination with another potent antibody, in my mind, have a great potential to truly be, much, much more effective. And we'll see that sort of synergy of going from 1 to 2 to 3, where we have many exponentially better coverage. So I do think that we need to make better antibodies. We need to have the right combinations that are targeting the viruses that are circulating, but continued characterization of the viruses that are circulating within an individual or within a population is also important to make sure we're using the right antibodies.
And I like the last question. Sorry that I don't want to take all. Can you give us some input about the activity of these VNAbs in non-B clades? Which data we have about that?
Yeah, that's a great question, and it's a huge sort of hot topic in the field right now. So we do have panels of viruses from around the world for which all of these antibodies have been tested, and we have a general sense as to for example, 3BNC and 101074, which are 2 of the antibodies that have been extensively tested in Europe and the United States. We think they have a little bit less general breadth and potency against clade C viruses and against clade A and D viruses, but we still think that that's what our virus panels tell us so far. But there are efforts, for example, from the AMP trial, in which we have multiple individuals who recently seroconverted. We can look at those viruses that established infection and test their sensitivities to these antibodies. And so we're getting more sort of real-time data about the viruses actually causing infection in various communities. And then we'll get a sense of how active these antibodies are. There are some antibodies being developed very specifically for clade Z viruses. So some of the V2 targeting antibodies like CAP256, which is really active against clade C viruses and not that active against clade B viruses. That's a great example of a virus really targeting that specific virus. But we need to continue to make sure that we understand the viruses that are circulating and have the antibody, have a sense of what those in vitro measures are. And then that's where we are kind of right now. And the concept of this small ATI for prevention study, where we ask if those antibodies can suppress people living with HIV in a switch study context, maybe that's enough information to inform our understanding of their ability to prevent circulating viruses.
Thank you.
Hi, this is Tom Giordano from Baylor. Wonderful talk. Thank you. To follow up on Roberto's questions a little bit, we've seen SARS-CoV-2 mutate quite rapidly to have escape from monoclonal antibodies and even combination monoclonal antibodies. In a broader sense, I know you're talking about some of that emergent resistance and innate resistance that some of the HIV has, but in a broader sense, do you think the field can actually get to a point where there are a couple products that would do this? How does that look? Do you think a patient would actually have to sort of rotate BNAbs periodically, or some other approach? Because there was subclinical replication in some of those participants in the clear study. They were replicating at low levels. So I'd love to hear just more in general your comments on that, what you think this could look like in 10 years.
Yeah. So first, the great question. And I think that the world has sort of been reawakened to the concept of virus evolution in the face of our SARS-CoV-2 and COVID pandemic. But it's important to remember that HIV actually replicates or mutates at a much faster pace and has a much greater diversity than anything we've seen, even with our continuing evolving variant situation in SARS-CoV-2. So we really have been dealing with virus evolution and rapid escape in HIV for a long, long time. You just have such a massive burden of people with replicating virus with COVID right now that's adding fuel to the fire. But to your points, first of all, in the eClear study, when we saw the virus replication, those were people off any therapy. So I didn't explain it very well, but they got their antibodies and their treatment interruption, their antiretrovirals. They were suppressed for about a year or two at least, and then they underwent a treatment interruption where they had basically no therapy. And off all medicine, then they had the virus come back a little bit, but be controlled by their own immune responses. And so that's this post-antibody effect that we're hoping is because it's improved our HIV-specific immune responses, potentially reduced that reservoir a little, but really ramped up our immune system's ability to handle that virus on its own. So that is a less concerning situation for resistance. However, all these other situations when we're using antibodies in the setting of, not with ART suppression, the potential for resistance is real. What I imagine as the best case scenario moving forward is that we identify several antibodies that are quite broad and quite potent. And with that combination, if they're used in these long-acting formulations where maybe you need to be dosed every 4 to 6 months and you keep very high levels, there's no chance for that virus to replicate. And so we won't see the evolution of new escape. And so as long as you don't have pre-existing resistance or you have very little pre-existing resistance, then you'll be able to have just full virus suppression on those antibodies, and they should be durable and long-lasting. And we wouldn't need to be cycling through multiple options. But that is an idealistic and optimistic interpretation of what we'll see in the next 5 to 10 years, I think.
Thank you. I guess the other point to make is that because SARS-CoV-2 is so more contagious than HIV, you don't expect the rapid evolution outside the individual patient to the same extent.
Yeah, the sort of are not the different the dynamics of the infection and the pandemic are really different. But you're right. I do think it's a good, way to let people understand, a wake-up call for all of us about how quickly viruses and pathogens in general can evolve. And, so many of our antibodies for SARS-CoV-2, which seemingly had a ton of potential, they're all basically rendered impotent right now because the virus has had the opportunity to go from person to person to person on such a large scale that even a relatively slow mutation rate enables escape on a population level.
We have one question here at Memorial Hermann. Oh, hey, this is Ben Barnett. So thank you for that. I've been in HIV field a long time, but this is all new to me. So thank you for that. I had a question kind of similar to Dr. Giordano because I'm trying to imagine how these would look in the clinic, say once the vision is realized. And now with the advent of long-acting antiretrovirals, would it be that these BNAbs you would envision that eventually, as you mentioned, patients would come off all therapy completely, and then they might have this low-level viremia or minimal viremia controlled by their own innate immune system? Or would they have to get continuously dosed every 6 months, every year, whatever, with BNAbs, and how that would be different than just the development of long-acting antiretrovirals when the patient still has to come in? Or is it a possibility that this would be combined somehow to do complete viral eradication and a virologic cure of HIV?
Yeah, that's a great question. I think you've kind of highlighted the sort of menu of options that are being tested right now. So much like long-acting antiretrovirals, long-acting BNAbs could be used either with long-acting retrovirals or as their own long-acting antiretroviral. And every 6 months, etc., you come in and get your infusion. And that antibody, which doesn't interact with any other, doesn't go through the same liver metabolism as small molecules, would be a potentially more friendly or less toxic version of a drug-based virus suppression. Another consideration would be maybe we find out that if we use the antibodies in the right way, we can slowly over time boost immune responses and chip away at the reservoir. And so maybe these antibodies are an antiretroviral, they're suppressing virus, but they're also working long-term to help for a cure strategy. Or maybe pie in the sky, we can do it faster, and BNAbs either by themselves or with another agent are part of a short, let's say 3 to 12 month therapy, which afterwards we think there's a good chance if we stop all therapies, virus will truly not come back. Now, your point about the fact that we have in that ECLAIR study, those folks had viremia, that's difficult for many HIV practitioners to imagine, given how far we've come with antiretrovirals and how important it is to suppress virus for personal and public health reasons. But maybe we can get to a point where the virus is very low level and we think it's not a risk for transmission and not a risk for disease progression. And that would be a way to allow people to be off therapies and with a sustained virologic remission type picture. But those are all theoretical at this point.
Then there's a question in the chat here. I'll read it off. Dr. Kamada says, can you comment on the host response to the BNAbs and rate of antibody clearance and how many times they can be given to a patient?
Yeah, that's a great question. So these antibodies are human antibodies, and when given by themselves, we've seen almost no anti-drug or anti-idiotype antibodies. So we've been able to give them over and over again without any sort of immune responses generating. Now, I will say that when they're given in, when they're given with some immunomodulators, so we've had some kind of like boosting agents or adjuvants given with them, and sometimes that can cause ADA, but when they're given by themselves, we have seen almost none of that. So it's obviously a concern when we think about monoclonals in many other contexts, but because these are human antibodies, we haven't seen that yet. Again, as we move into more engineered antibodies or antibodies with unique functions or sort of adaptations, that may not be as straightforward because every time we stray from these natural antibodies, we increase the chance that the body recognizes it as foreign and may develop a response against it. But as of right now, these fully human antibodies are extremely well tolerated and can be given over and over and over again.
All right. If there are no more questions, thank you so much, Dr. Barr, for speaking today. And for those of you that did not get a chance to sign in, let me go ahead and put the information in the chat just one more time so that you can get your credit, and please complete any evaluations and post-tests available. And I hope you all have a good rest of your day. Thank you.
Thank you very much.